All right, welcome everyone to the lecture on meiosis and sexual reproduction. Our objectives with this lecture are to explain the stages of meiotic cell division. We'll explain the role of sexual reproduction in genetic diversity, and we'll also explain how chromosomal abnormalities can occur by meiosis. First, let's talk about genes. Genes are segments of DNA that code for the basic units of heredity. Genes code for the production of RNAs and polypeptides. Offspring acquire their genes from their parents by inheriting chromosomes. There are two major types of reproduction: asexual and sexual reproduction. Asexual reproduction produces clones or genetically identical offspring to their parents. Meiosis produces gametes or sex cells like sperm and egg. Asexual reproduction requires just one parent, whereas sexual reproduction requires two parents of opposite sex. Asexual reproduction produces very little genetic variation in populations. Genetic variation occurs only through mutation. Sexual reproduction has comparatively high genetic diversity. Asexual reproduction is fast and energy efficient. They don't have to spend time going to find and impress a mate. Sexual reproduction is slower and it consumes a lot of energy. An example of asexual reproduction is budding or binary fission. Sexual reproduction, I think we're mostly more familiar with. This is how humans reproduce in trees. Let's look for some imagery here. So this is a really nice image of a bud. You can see there's a tiny offspring that looks just like the parent here growing off the side. And eventually, when this bud is fully formed enough, it will detach and become its own independent individual. This is a hydra. You can find them in Michigan. They live on the bottom sides of lily pads. They're related to jellyfish. Trees, on the other hand, we know have to be pollinated, and then they produce a seed, and that's how they reproduce sexually. All right, so let's talk a little more about chromosomes. Chromosomes in your somatic cells, the cells that make up most of your body, are diploid. That means they have two copies of each chromosome, one from your mom and one from their dad. And for humans, that makes a total of 46 chromosomes. Every species has a different number of chromosomes. Each pair of homologous chromosomes includes one chromosome from each parent. Autosomes, we have 22 pairs of chromosomes that do not determine biological sex. And if you do your math, right, 22 times 2 is 44. That means that there's one chromosome that does determine sex, biological sex, and those are the X and Y chromosomes in humans and most mammals. So gametes are haploid. They have just one copy of each chromosome. And in humans, that means there's a total of 23 chromosomes made up of 22 autosomes plus one sex chromosome in every gamete, in every sperm, and in every egg. And when those two things unite to form a zygote, that's when we end up with a diploid cell. And that's literally where you get your one copy of each chromosome from mom and one copy of each chromosome from dad. Apparently these are out of order. Sorry. So in humans, most biological humans who are female are XX. Most biological males who are human are XY. But of course, like all things in biology, there are general patterns, but this does not include everyone, which is part of what makes biology so fun and interesting. All right. So if we jump back down here to gametes, an egg has 22 autosomal chromosomes plus an X. A sperm can have 22 autosomes no matter what. And then it either carries the X chromosome that the father got from his mother, or the Y chromosome that the father got from his father. So it's the sperm that determines what the biological sex of the zygote will be. All right, homologous chromosomes in a somatic cell. So just a regular body cell. In this cell, we have a diploid cell, 2N, and the total number of chromosomes equals 6. So, where's my cursor? There we go. Sister chromatids of one duplicated chromosome are represented here. So this cell is preparing to divide, and it's copied each of its 6 chromosomes, right? 1, 2, 3, 4, 5, 6. These are sister chromatids because they've been copied. These are both paternal chromosomes from dad. And then over here, we see the homologous chromosomes. These would also be chromosome number 1, right? Because these are the biggest. And it's also been copied, and these are the maternal chromosome number 1s. We also have 2 non-sister chromatids here in a homologous pair. Make sure that you understand the difference between homologous chromosomes, meaning chromosome number 1 from mom and chromosome number 1 from dad. To be able to differentiate between those homologous chromosomes here, like red and blue, that are the same size, versus sister chromatids of a single duplicated chromosome. When a cell's getting ready to divide, remember it copies its DNA. So make sure you understand the difference in those vocabulary terms. Good. And this would be a great place to pause the video and define homologous chromosomes. All right, karyotype. This is a picture of an organism's complete set of chromosomes. So it's a pair of homologous duplicated chromosomes. So we have sister chromatids here together on the right, and sister chromatids here together on the left. So this would be the duplicated copy from one parent and the duplicated copy from the other parent. Anyways, we can sort them by size, and that's how we identify and name the chromosomes. We pair them up from biggest to smallest. The biggest chromosomes we call chromosome number 1. The next biggest are chromosome number 2. The next biggest are chromosome number 3 and 4, and so on. And then we always list the sex chromosomes last. All right, making a karyotype. So this is what it looks like before those chromosomes are sorted. And then after they've been sorted, we have 22 pairs of autosomes, 1 through 22, plus 1 pair of sex chromosomes. So this individual has 2 copies of the X chromosome and 0 copies of the Y. Is this individual biologically male or female? Good, female, right? 1 X from mom and 1 X from dad. All right, how about this karyotype? Is this individual biologically male or female? Good, X and Y. So this karyotype is biologically male. All right, karyotypes can also be used to identify genetic abnormalities. All right, and if you look as we go through, see if you can spot the abnormality in this karyotype. 2 pairs, 2 pairs, 2 pairs, all the way down the list. 2 pairs of every chromosome. They all look very similar until we get here. There are 3 copies of chromosome number 21 here. All right, and this individual has a condition called trisomy 21 or Down syndrome. Down syndrome is trisomy 21 is one of the only genetic conditions where you can have an extra set of chromosomes that's not lethal. So the reason why we don't see people with trisomy chromosome 2 is because that's a lethal genetic condition. Zygotes that have 3 copies of any other chromosome, apart from 21 or the sex chromosomes, can't survive. So those individuals are never born. All right, cancer cells, and this is really interesting. Some cancer cells have abnormal numbers of chromosomes as well. So if you look at this karyotype from a metastatic melanoma, so it would be a melanoma is a skin cancer. Metastatic means that it has traveled from its point of origin to cause cancer in other parts of the body as well. It's got 3 copies of chromosome number 1, 1, 2, 3, 4, 5, 6 copies of chromosome 2. It's just a really unusual looking karyotype. And it's not surprising that cancer cells behave in ways that regular cells do not, right? Well, interesting things to think about. Here's the karyotype of breast cancer cell. All right, HeLa cells. So this is one of the oldest and most commonly used human cell lines in research. They are cervical cancer cells taken from a woman named Henrietta Lacks, hence the name HeLa. Henrietta Lacks died in 1951, not too long after her cells were collected. She did not consent to have these cells taken and used for research purposes. Now, what makes these cells so special is that they're immortal. These cells don't die after a few cell divisions. They have an active version of the enzyme telomerase. And they've been used in research to do incredible things. The vaccine for polio was developed using Henrietta Lacks cells. Cancer, AIDS, virus, and radiation research have all benefited from the use of the HeLa cell line. And it's estimated that the cells produced in culture by researchers have exceeded the number of cells in Henrietta's own body. Here's a karyotype of the HeLa cells. So HeLa cells have aided scientific advancement in human medicine, but it's come at a cost, right? So tissue was taken in a biopsy from Henrietta Lacks without her knowledge in 1951, the year she died. In 1953, 2 years later, Jonas Salk was supplied with HeLa cells from a cell culture factory established at Tuskegee University. That's less than a year later. Salk's polio vaccine was ready for human trials. In 1955, Theodore Puck and Philip Marcus at the University of Colorado at Denver successfully cloned the first human cells using Henrietta Lacks cell line. In 1966, scientists injected HeLa cells into unwitting test subjects to study how cancer spreads, prompting the National Institutes of Health to establish medical internal review boards and informed consent. In 1989, a German virologist used the HeLa cells to show that human papillomavirus causes cancer, a discovery that would land him a Nobel Prize. In 2013, the complete HeLa cell genome was sequenced and published without the Lacks family's knowledge. Also in 2013, the National Institute of Health Director Francis Collins announced a policy of controlled access to the cell line genome based on an agreement reached after several meetings with the Lacks family. So, like all things, we need to make sure that we pause to think about what we're doing in science and make sure that what we're doing is moral and just. So people make mistakes, but I'd like you all to pause and think about the cost to Henrietta Lacks and her family. How would you feel if your mother or grandmother's genome were published, one of the first human genomes published, without your family's knowledge or consent? How would you feel about your cells being taken from you and used by researchers around the planet without your knowledge or consent? These are not small questions. So take a moment to think about that. We've come a long way with developing ethics review boards and trying to hold ourselves as a scientific community accountable for doing our work in ethical ways. But this is a really important thing to think about, and something every scientist and every human who votes to have at the back of their mind. What's the cost? All right, so there's a lot of controversy around this, obviously. Since her cells were harvested without her consent, the discarded tissues can be commercialized and sold for profit. The genome was published without the family's consent. And there's a really interesting book, if this is something that you'd like to learn more about, called The Immortal Life of Henrietta Lacks. All right, and here's a picture of her headstone. In loving memory of a phenomenal woman, wife, and mother who touched the lives of many, here lies Henrietta Lacks, Gila. Her immortal cells will continue to help mankind forever. Eternal love and admiration from your family. All right. So let's move on to something a little lighter. Life cycles. Reproductive history of an organism from conception to the production of their own offspring. Fertilization and meiosis alternate in sexual life cycles. Meiosis is cell division that reduces the number of chromosomes. Remember, we go from diploid to haploid gametes. And this is where sperm and eggs are created. Fertilization is when gametes are combined, when sperm and egg unite. The fertilized egg is called a zygote, and this is a diploid cell. Zygote divides by mitosis, regular cell division, to make a multicellular diploid organism. There are varieties of sexual life cycles. So here we see animals where we have haploid gametes that come together, right? Two haploid gametes are fertilized to produce a diploid zygote, which undergoes mitosis to develop into a diploid multicellular organism, which does meiosis of its own to produce gametes. It finds a mate, those gametes unite and fertilize to create a new offspring. And so the cycle continues. Plants and some algae use a haploid multicellular organism, gametophyte. So they alternate between having a haploid part of their life cycle and a diploid part of the life cycle. And this is a little different than the way animals do it. So if we start here where we have our diploid multicellular organism called a sporophyte, it undergoes meiosis and produces spores. Those spores produce gametes. When two gametes unite, they're fertilized. But in between here, the spores grow into a larger haploid cell. This haploid multicellular organism is called a gametophyte. So that gametophyte produces gametes. The gametes unite with another individual, and it creates a fertilized zygote, which develops into a diploid multicellular organism. So it's almost like there are two different organisms to get you to sexual reproductive event. So the sporophyte here is diploid, and then the gametophyte that's produced up here is haploid. All right. Lastly, we have the way that most fungi and some protists reproduce. There's a haploid unicellular or multicellular organism, just like the gametophyte. It does mitosis to grow, just like the gametophyte. It produces gametes, which become fertilized, and it grows a diploid zygote. That zygote undergoes meiosis to produce haploid gametes, and those haploid gametes are what develop into a unicellular or multicellular organism. So the zygote down here doesn't do mitosis. That's the difference for fungi and protists. All right. Oh, we've already talked through these. All right, so meiosis causes a reduction each time it divides. There's a reduction in the total number of chromosomes in the cell. So we've talked about the cell cycle and mitosis. Cells spend most of their lifetime in interphase, right? We have a pair of homologous chromosomes in a diploid parent cell. When the cell prepares to divide, they duplicate. They duplicate their chromosomes, right? So now we have sister chromatids. And in meiosis 1, it behaves in a lot of the same way as mitotic cell division. We have homologous chromosomes that are separated. So the chromosomes from mom go to one cell, the chromosomes from dad go to another. And then a second round of cell division occurs. This is different from mitosis. In meiosis 2, haploid cells are produced as those duplicated chromosomes, the sister chromatids, are separated. So after meiosis occurs from a single parent cell, we should have 4 haploid cells. So 2 eggs. Sorry, I guess these would be the eggs since these are... No, not necessarily. Anyways, we end up with 4 haploid gametes, whether they're eggs or sperm. Depends on which parent this first cell came from, right? All right, so the cells divide twice. The result is 4 daughter cells, each with half as many chromosomes as the parent cell started with. Oops, sorry, too far. So meiosis 1 separates the homologous chromosomes. So we have prophase 1, where those homologous chromosomes are becoming visible. Metaphase 1, they all line up at the equator of the cell, just like mitosis. We've got the spindle fibers connected to the centrosomes. All right, the sister chromatids remain attached as they separate. But there's something special that happened here. Look, what do you notice? Once we get to anaphase 1, the chromosomes aren't solid color anymore. What happened? Well, when the chromosomes line up at prophase 1, something called crossing over occurs. And where these chromosomes overlap, they swap chunks. So you end up with unique genetic conditions that you didn't see in the parent, right? Neither parent has a chromosome that's blue and red. So we get unique genetic variation that occurs during crossing over. in prophase 1 when these chromosomes line up, the pairs of sister chromatids, and they swap chunks of DNA at the chiasmata. And this is important because the cells that arise from meiosis can be genetically distinct and unique, variation that wasn't found in either parent, which is huge. All right, so interphase, the chromosomes are replicated. Prophase 1, we have synapsis, where the homologous chromosomes pair up. They form a tetrad with the 4 sister chromatids, right? Crossing over occurs at the chiasmata. That's where they touch. See where they overlap? And where they overlap, they swap sides. So this piece actually jumps over and attaches to this chromosome. And this piece jumps over and attaches to this one. In metaphase 1, the tetrads all line up. Anaphase, those pairs of homologous chromosomes separate. The sister chromatids are still attached by a centromere, however, so they go together. And in telophase 1 and cytokinesis, we end up with 2 haploid cells. Each chromosome equals 2 sister chromatids at this point. In some species, the chromatin and the nucleus reform. All right, now we're ready for meiosis 2 down here. So we start with one of these haploid cells. We'll carry it down here to prophase 1. They'll line up on the equator of the cell for metaphase 2. The sister chromatids separate now, and we have haploid daughter cells that form after telophase and cytokinesis. All together, from a single parent cell, we should end up with 4 haploid gamete cells. Because this will happen with meiosis 2 happens with each of these 2 daughter cells. So that'll produce a total of 4. In prophase 2, there is no interphase. There's no crossing over. The spindles form. In metaphase 2, the chromosomes line up in the center of the cell. In anaphase 2, those sister chromatids are separated. Telophase 2, we have 4 haploid cells. The nuclei reappear at that point, and each daughter cell is genetically unique, different from the parent cell. So there are 3 ways that meiosis is different than mitosis. Prophase 1, synapsis and crossing over. That's something unique to meiosis. Metaphase 1, pairs of homologous chromosomes line up on the metaphase plate at the equator of the cell. 3, anaphase 1, homologous pairs separate, but the sister chromatids are still attached at the centromere at this point. All right, and here's a nice summary slide to compare and contrast mitosis versus meiosis. The mitosis is regular somatic cell division. We should end up with daughter cells that have the same genetic complement as the parent cell. Meiosis, we have genetic variation that's produced by crossing over. And there's also random assortment of which homologs go where. We don't have all blues and reds moving together, right? We have combinations. Good, and the end result is 4 haploid cells. All right, there is no crossing over that happens in mitosis. That's unique to meiosis. And the roles of these 2 processes are very different. So the purpose of mitosis is to enable growth, produce new cells for tissue repair, and in some species, mitosis is used for asexual reproduction. Meiosis is used for the production of gametes or spores. So gametes in animals, spores in plants. It reduces the number of chromosomes by half, and it introduces new genetic variability among the gametes and spores. All right, so the 3 sources of genetic variation, and you should know these for the exam. Crossing over is the exchange of genetic material. We call those chromosomes that are mixed, red and blue, recombinant chromosomes. They've been recombined to create new chromosomes. Next is the independent assortment of the chromosomes. So if we've got 2 different chromosomes, chromosome 1 and chromosome 2, they could separate where the 2 from dad go together and the 2 from mom go together, but it's equally likely that they won't. It's equally probable that 1 from dad and 1 from mom will separate together, and the opposites here. So you could have gametes like this, or the same cell could produce gametes like this. All are equally likely, and this just increases the amount of genetic variation available. Random orientation of homologous pairs in metaphase 1 is responsible for this. All right, and the last source of genetic variation is random fertilization. So this is a micrograph, an image taken under microscopy of an egg cell being surrounded by sperm. And here you can see the moment of fertilization as the sperm head burrows through the outer layer of the egg. Once this occurs, the egg produces kind of a shield that prevents more than that one sperm from fertilizing the egg. So any sperm and any egg. And that's another randomness to the whole process as well that produces and encourages genetic variation. So it's 8 million times 8 million. These are all the different genetic possibilities for sperm from a single father. And these are all the different possibilities for eggs from a single mother. That means any one couple can have 64 trillion different genetically distinct children. All right, so mitosis and meiosis, both are divisions of the cell nucleus. Mitosis is just for your regular body cells, somatic cells. Meiosis produces gametes, sperms, eggs, spores. Mitosis has just one cell division event. Meiosis, cells divide twice. Mitosis produces 2 diploid daughter cells. Meiosis produces 4 haploid daughter cells. Mitosis produces clones. The daughter cells are genetically identical to the parent. Meiosis produces genetically different gamete cells. So less than 1 in 8 million are exactly alike. Mitosis is how our cells divide from the zygote stage until we die. Meiosis occurs in females before we're even born. Follicles are formed. Mature ova are released beginning in puberty, but those ova have already been produced by meiosis before newborn baby girls are even born. All right, mitosis, the purpose is generally growth and repair. The purpose of meiosis is for reproduction. Mitosis does not have synapsis and crossing over. All right, I think we've already seen these images earlier. So by now, you should be familiar with meiosis and genetic variation. You should be able to explain the stages of meiotic cell division, explain the role of sexual reproduction in genetic diversity, and explain how chromosomal abnormality can occur by meiosis. Oops. As always, if you have any questions, write them down on your slides sheets. You can bring your questions to student office hours or to class, and we'll chat about them then. Thank you for watching.